I'm delighted to be able to hear this talk and introduce Elizabeth Clare. She got her PhD from the University of Guelph, then did postdocs at Bristol and Cardiff universities, and worked in a faculty position at Queen Mary University in London for 8 years before moving to York University in 2021. And she is interested in biodiversity at all levels and all aspects, how it's changing, how it's affected by landscape, by diet, by climate, by interactions with other species, and how to measure it precisely in novel ways. So she measures DNA in water to get at the biodiversity of fishes. She measures DNA in the gut of leeches as an indirect measure of biodiversity in vertebrates. She measures DNA in feces to figure out who's the predator for whom and how far seeds are dispersed. And in a recent breakthrough we'll hear about today, she's figured out how to measure it in the air and figure out what kind of mammals are nearby, even ones in caves or underwater. She's also discovered that in the air samples that have been taken around the world forever and saved, there's useful data on geographic variability and historic variability, and she's been mining that treasure trove. This was a major discovery, the airborne DNA. You may have heard about it on NPR, CNN, CTV, read about it in The Scientist, The Economist, The Financial Times, the Smithsonian Magazine, or elsewhere. She does a lot of lab work, but she really also loves field work. She's taught field courses in the desert in Africa, in the South African savanna, in the Costa Rican rainforest. She maintains a mobile DNA lab in Belize, where she's studying biodiversity in bats, and there's an annual field trip there every year for her lab. It looks like from the pictures, they do a lot of good science and have fun too. Her work is published in the most prestigious scientific journals, including recently Nature and Current Biology. And just this last fall, she was elected to the Royal Society of Canada in the College of New Scholars. And that's how we found her, because we looked at that list, like who's in the area, who's doing really interesting work. And there she was. And she said, yes, she'd come and tell us about all this fascinating research. So we really look forward to this talk. And thank you very much, Elizabeth.
I'm going to try and make that bar go away by telling, oh, it's saying it's okay. I'll turn it back. Okay, we back? Right. Okay, thank you very much for the invitation. There's nothing any academic likes more than talking about what they do. So I like getting invited to tell stories. I have moved around quite a lot. I'm from Ontario. Most of my adult life was lived in the UK. We moved back a couple of years ago, and I've been having a lot of fun because what we're doing has caught people's attention. I'm going to start, though, with a really simple question. And I now start all of my public lectures with this because I was talking to someone on the radio, and the interviewer asked me, well, why should we care about biodiversity? And it was so obvious to me that I made a complete mess of the answer, because I couldn't believe somebody wouldn't know that already. It just, to me, it's intuitively obvious. And so I said something about bees and pollination and then moved on quickly because I couldn't think how to actually answer the question of why I care about biodiversity. And so I sat down for the purposes of a grant application, and I made a list of the reasons why you should care about biodiversity. So we'll start with the one where I kind of flubbed through the answer, which is food security. There is huge evidence that biodiversity influences the productivity of our landscapes. Soil, microbial, fungal biodiversity has a direct implication on the amount of food you can grow. Pollinator biodiversity helps increase the variety of things out there for you to eat. So if you care at all about the reliability of our food production system, biodiversity matters. Clean water. There's still no better way to clean your water for consumption than to have a good functioning marshland system that can clean it and filter it. Better than any biological, sort of chemical, technological filter we created is to let biodiversity do it for you. It's great at removing heavy metals and at taking toxins out of water. Climate, There's a marvelous set of studies going on right now at McGill about the biodiversity of cities reducing the heat island effect. So cities are hotter than rural areas. You'll know that in downtown Toronto. It gets much hotter here than if you drove 100 kilometers out into the rural area in August. If you increase the biodiversity just of the trees, you can lower the temperature of a city. And in an age of climate change, that matters. My neighbor 2 doors away has no trees on her property. I have tons of trees. My property is cooler than hers, a few meters away. Biodiversity can regulate your climate. There's wonderful examples of how it moderates even the physical landscape. So a very classic example in ecology in Yellowstone Park, when wolves were reintroduced to the system, they completely regulated the biodiversity of everything living in Yellowstone Park. It had an actual effect on the physical geography, changing the courses of rivers, just through increasing and restabilizing biodiversity. So physical landscapes are altered by biodiversity. Pharmaceutical industries rely on what we call bioprospecting, going out and looking for new plants and animals and fungi with new biological compounds, which make it into things like medicines. The UN released a report on the cultural impacts of biodiversity, that so many cultural practices are based around knowledge and exposure to biodiverse areas, that it's part of our human culture to respect biodiversity. Art, if you Google biodiversity and art, you will have an endless set of pictures to look at, people who are influenced by what they see around them in the natural world. Human health, your actual physical health is influenced by your exposure to more complex green spaces. And there are really, really good studies that link the propensity of people to be more resilient to microbes and infections just for their exposure to more natural landscapes. And human mental health is massively influenced by your exposure to more complex green spaces and biodiversity. So all of these things matter in sense of trying to preserve more complex systems. Just exposure to one sort of green field is not as good as a complex landscape. So then what is biodiversity? And that's the next question is, if you care about it, what is it, this thing we're supposed to care about? So there's different ways of looking at it. In terms of the diversity of things that we think are out there, the names we put on stuff, most of them are insects. So we have named more insects, arthropods, than anything else. And this is a sort of scale diagram of what we have names for. With the animals, their sizes are adjusted to the diversity of things that we know about. And we can contrast that to what probably exists. It's a rebalanced scale. Things like the worms. There's probably a lot more worms than we know about out there, the annelids. So we haven't really named things in proportion to how they really exist. There are tons and tons and tons of things that we have never seen before. And one quite sort of provocative thing that Forbes put out a while ago is that ugly things deserve our attention. So you hear a lot about wanting to protect panda bears and cute and cuddly stuff. What we really need to think about when we talk about biodiversity are what one of my friends refers to as the little things that make ecosystems function. And ugly stuff deserves our attention as well. But that's not what we do. So even as scientists, we aren't very good at this. This is a diagram taken from the Global Biodiversity Information System of the amount of things that are recorded. The pink is birds. So if you believed what we've actually reported in these sort of databases, the world is composed of birds and a bit of other stuff. Okay, it's not how it really works. What we do know is that it's in decline. We don't really know how much, but we know there are declines. This is a graphic that was created by The Guardian, a newspaper in the UK a few years ago. And what it suggests is that since the rise of human civilization, so we're talking tens of thousands of years, the number of individuals that have disappeared, not species, but actual individuals, is what we should maybe be concentrating on. Roughly 83% of individual wild mammals are probably gone. Not the species, but the individuals. About 15% of the individual fish have disappeared. And what have we replaced them with? We talk about biodiversity declining on a global rate, this being a crisis. What does that mean? If we were actually to look at the number of mammals you and me out there, there's about 6,500 species of mammals, the rodents and the bats and the people and the sheep and the goats. If we just looked at their biomass, 60% of them are livestock, cows, and roughly 36% are us. Less than 4% of the biomass of mammals is wildlife. 96% of it is us and the stuff we grow to eat, and some dogs. So this is not the loss of species, but a complete rebalancing of how this system works. If you looked at birds, that's one. Okay. Birds, there's about 13,000 species of bird. 70% of them are chickens. That's what we've done. So very few of them are actually wildlife. Okay? Domestic wild thing, domestic stuff, chickens, cows, dogs, they have taken over the biomass of the planet. That's what we're really, really good at, is altering the balance. We can't see it in numbers of species yet. We haven't lost a whole lot of species, but we have utterly adjusted how they are distributed around the landscape. So the first sort of problem we have in biodiversity science is we have no idea how to measure any of this stuff. About 90 to 95% of the things that are living on Earth right now, we do not know about. We have never recorded them. There is no name for them. If you brought one to a specialist in that field, they would not know what it was. And that usually surprises people. It's really easy to describe a new species. So 90% of life is not known to science. Forget the stuff that's gone extinct in fossil record. Just the stuff alive now. Most of it we've never seen. Why? Because we have no infrastructure to actually monitor it. If you're an astronomer, you've got gigantic arrays of things based around monitoring space. We have no equivalent to monitor life on Earth. There is no system set up to actually monitor it. It's usually people like me out in a forest catching stuff and trying to record it. But you never go back and do it again. You only learn about the thing you're looking for. I can go and catch bats in Belize, which I do every single year, and tell you how the bats in Belize have changed from one year to the next. But while I'm there, I don't learn about anything else. I don't learn about birds or other mammals because the techniques we use are very, very specific and labor intensive. So, we don't really have a way of measuring stuff at large scales. Now, to switch entirely to what I actually work on, it's this stuff called environmental DNA. It is not new. It's about 20 to 30 years old in terms of our knowledge of its existence. It began really in the 80s with one woman who was surveying mud from microbes. And it was kind of forgotten about. And then it jumped ahead to the late 90s where it became a tool. So what is it? All organisms shed DNA. You are doing it right now. So where you're sitting, when you leave, we could come and pick up a trace of your DNA left on the chair. You just do this. You can't stop it. So it's bits of dead skin cells and some hair and saliva and anything that you leave behind. We call this environmental material, environmental DNA. And with enough money, I could actually come in here and I could sequence your chairs and work out where you all sat. Don't worry, I won't. But we refer to this collectively as environmental DNA, and all organisms shed it all the time. Whether you're a microbe or a mammal or a tree, you are shedding little bits continuously. It's how your body works. You can't stop it. But what we learned over the last 20 years is you can collect it. And you can learn stuff from this material. So it's just material shed and left behind. This is where we become forensic biologists. We're like crime solvers at the same time. It was really first popularized by a man named Eske Willerslev in Cambridge University in the UK. In his previous life, if I get his biography right, he had been a Siberian trapper before he decided to become an academic. And he went back to graduate school and he wanted to sequence DNA from things like mammoths that had been found in ice core, like in frozen in the permafrost. And no one would give him any. But what they gave him was ice cores. And so he had this crazy idea that he could find DNA from them in the ice, because DNA preserves pretty well when it's frozen. And so he began basically trying to get DNA out of something which looked like a block of ice. And you can do this. It works reasonably well. He worked on ice cores, and he tells a story that it was Christmas morning, and he got his first results back, and he had mammoth DNA frozen in ice core, and there was no one to tell because it was Christmas. And he went on to study things like sediment cores, mud cores. He got DNA from extinct giant moas, the birds. He reconstructed ancient landscapes of DNA that had basically been frozen and preserved for hundreds of thousands of years. So the animals are gone, but they've left behind this forensic trace, the same stuff you're leaving on the chairs. And since then, it's become this incredible tool in biology. So this is a picture of one of my students, Alejandro. He is a Mexican citizen, and he worked in these little remnant pieces of river. This is all that's left of the lake that really used to be Mexico City. So Mexico City is really built on top of an old wetland. And in here is the only place you can find the wild axolotl, which is this little thing. You get them in the pet trade. They never turn into fully adult things. They keep these juvenile characteristics. People keep them as pets. I've got my pointer now. And they're very, very cute, but they may be extinct in the wild. They're really hard to catch. They only live in this one sort of river system, and you cannot survey this. It's too destroyed and ruined. So what he went and did was he filtered water to see if he could find the DNA of the axolotl floating around. We do this all the time with fish. It's a great technique to survey fish communities. You literally use a filter and filter their DNA from the water. You never have to see the animal. It works really well. It's like making coffee. So if you're making coffee, you pour water through the grounds. The grounds get caught on the filter and the water goes through. It's exactly the same thing. We run the water through a filter. The DNA is caught on the filter and the water goes through. And now you've got a DNA sample. You can do this relatively easily. So Alejandro is doing this to try and see if he can find remnants of axolotls still in the wild. The net outcome is probably not. They're probably extinct in the wild. But since water became the thing people use to find this kind of material, it's really expanded into huge other areas. You get environmental DNA in soil samples. You can dig down and learn about all the subterranean life through a soil sample. Water is the famous one. This is actually a picture that I've taken from the US Geological Service, where this is just a tool they use to go out and measure waterways. Honey, you eat honey, you're eating the DNA of the plants and the insects at the same time. Honey is a great conveyor of DNA. You can learn where the bees were pollinated or were pollinating, which plants they were visiting from a scoop of honey. You don't have to go and observe the bees. Just get the honey and you can record where they've been because the DNA is left behind like this little footprint. Rain washes DNA off leaves in the forest. You can collect the rain and filter it just like river water. This is a mosquito sucking on a friend's arm. The blood in the mosquito can tell you where the mosquito has been. You can track pests through their diet and literally footprints. So we talk about eDNA like a footprint left behind after you leave. You can literally get DNA out of footprints in the snow to see who left the footprint. And we found it in lots of places, surfaces of leaves. If you collect a whole bunch of insects in a trap, they will carry the DNA of things they have encountered in the few days before you caught them. Even inanimate stuff like bits of rock are great at attaching DNA. DNA is kind of sticky, it's charged, and so it will attach itself to inorganic particles. And we've been doing this for a long time. One of the first fields I worked in was diet analysis. So just the idea that a predator eats a lot of stuff, its gut is packed full of DNA. It's feces. I was the queen of feces for a long time. You would not believe what Canada Post has brought me in the mail. There was a few years where all of my mail was feces. People would send me stuff. Could you analyze this? Because it's full of DNA from whatever they ate. And we were doing things like tracking the movement patterns of colonies of bats based on what was in their feces. This is bat feces, bat guano crushed up under a microscope. Those are fragments of insect. All of them have DNA attached to them. So we could work out everything they ate. And once you know what they ate, you can work out where they were hunting, what the landscape was like. So we were doing ecology, having never seen the ecosystem, the animal, or the prey, just from what they left behind. It's like going through your garbage pail to figure out where you're shopping. I could work out your favorite grocery store from what's in your garbage. Same thing. We moved on in Southeast Asia. One of my graduate students, Rosie Drinkwater, worked on a project using leeches to survey mammals. Leeches are really easy to catch in Southeast Asia. These are terrestrial leeches. All you have to do is walk into the forest and stand still and they will find you. They crawl out of the leaf, you can scoop them up, bring them home, and get the DNA out of their gut. And you can find out all the mammals that they have been feeding on from the DNA left inside. And Rosie was able to track bears and sun bears and elephants, wild pigs. This is a pangolin. This is one of the most trafficked animals in the world. If I had sent her to Southeast Asia to find me evidence of pangolins, she probably would have failed. I sent her to find leeches, and the leeches found the pangolins. It's like having an army of free field assistants. She got the idea to use dung beetles. These are famous little beetles that roll mammal dung around. And as they roll the dung around, they become completely covered in the DNA of the animal that left the little sample behind. And they're even easier to catch. You dig a hole, you find a friend who's willing to give you a sample, suspend it over the hole, and the beetles fall in. Come by the next day and you have a trap full of beetles. Pull them out, swab their backs to get DNA off them and sequence it. And you can learn about all the animals they've encountered. In this case, banded civets, a really rare mammal. She found their DNA on some dung beetles. So you go after the thing that is really good at finding the stuff you're interested in. So the intermediate. So this is the kind of stuff we were working on, using armies of dung beetles and leeches and filtering water and all of these sort of things to learn about biodiversity, to learn about vertebrates, fish, axolotls, mammals, things out there in the wild that we couldn't actually find because they're too hard to capture. Now, we're going to play a game, just a little one. Everybody take a deep breath. You'll never think about that the same way again if you play along. So while we were doing this, about 4 years ago, I got asked, I was living in England, and the chief scientist group of the UK commissioned 5 articles on how we can use DNA to study ecosystems. And I got asked to write about land. So I was assigned terrestrial. As part of this group of articles they commissioned called Understanding Ecosystem Resilience. The UK is very, very ahead of most places in using DNA as a tool to help preserve and learn about their own ecosystems. And so I wrote this report for them about all the dung beetles and leeches and soils and stuff, and how you can collect DNA in the wild. And I said, you should filter the air. I assumed that people had done this. I mean, this is pre-COVID, so we weren't really thinking about stuff floating in the air all that much, but I just assumed that given that pollen moves in the air and fungal spores, that this would be a done thing. And I was trying to find case studies to explain how we could do this, and there weren't any. And so I said, well, we should look in the air. And one of those humiliating, put your money where your mouth is kind of moments, somebody asked me to go and try it. So what we knew at the time, this is less than 4 years ago, was there were really very little known about whether DNA could move through the air. You would think it's obvious now, thinking about viruses and microbes that transmit through the air, that this should have been obvious, but it wasn't. It was a primarily theoretical field if you weren't interested in epidemiology and the movement of disease. Nobody thought you could track an animal in the air. We think of the air, we say thin air. We think of the air as empty, and it is anything but empty. There was one study by a friend of mine, Mark Johnson, who had looked at plants, which seems reasonably obvious. Pollen travels in the air, so you should be able to find plant DNA in the air. There was one study from Hawaii about fungus in the air. That was it. I couldn't find anything else. Simultaneously and nice and coincidentally, at the time, my university offered a small amount of funding for what they basically called crazy ideas. Things that were too crazy to write a grant. Nobody was going to give you money for that. They said, we expect most of these projects to fail. If something works, it could change everything. That's what they wanted. They wanted the craziest idea you could come up with. And I said, I'd like to vacuum the sky. It took a bit of convincing. But this is the experiment we came up with. These are naked mole rats. They're marvelous little things. They live in captive colonies in lots of universities because they don't appear to age particularly well. They're not subject to things like cancers. They're very, very resilient to stuff that we worry about. So they're wonderful study models. We happen to have a bunch of colonies of them in the lab. And so I said, well, let's back off a little bit from vacuuming the sky. Let's just try to suck the air out of the colony and see if there's naked mole rat DNA in the air. So this is a little experiment. This is a filter pulling air out of the little colony. We took a sample of air from the colony and a sample from the room. My expectation was the room is empty. The colony might have DNA from the mole rats. I was wrong in the most spectacular way. I was right, but everything had DNA. The room was crammed full of DNA. People DNA, mole rat DNA, DNA from a dog, which we eventually tracked to the guy who feeds the mole rats, walks his mother's dog on the weekend. And on Monday morning, he tracked the DNA into the room. It became aerosolized, and we would catch it. And people then usually ask, well, how do you figure out what the DNA is from? The best way to think of it is like a game of Go Fish. You have some unknown DNA. We have large databases of known DNA, and you play a game of fish. I have this unknown, you have a bunch of stuff you've stuck in a database that's known. Can I find a match? And for common stuff, the answer is usually yes these days. So I have an unknown, I match it to your known, I now know what my unknown is. That's how we play this game. Not really much more difficult than that. So we had this little experiment. We published it in PeerJ, tiny little, one of my favorite little journals for fun stuff you've tried. I published there a few times. It's a great little open access journal, and people got really, really interested. They thought this was really fun. So we moved on to another experiment. COVID hit. We couldn't go anywhere and couldn't do anything very exciting. This was within the allowed driving distance of my house in the UK, which was you were allowed to go about 15 kilometers from your home at the time by law during COVID lockdowns. We were really, really restricted in the UK. This is a zoo. It's a zoological park called Hamerton, and they was shut down, but the animals had to be fed. So they allowed us to come and walk around the zoo taking air samples. And the reason zoos are really interesting is that they're basically a captive group of non-native species. If I take an air sample and I find a cow, it could be that cow or someone's dinner or a cow from 300 miles away, I don't know. But if I'm in rural England and I detect tiger, there aren't that many sources. So zoos become really useful because they allow you to find the stuff that's unusual. And so we walked around the zoo taking air samples. So we knew a complete map of where everything was. Nothing moves around. It's a zoo. They're all in their own little enclosures. We took air samples inside their enclosures, a bunch of dingoes watching me, take an air sample from them. We did them outside where they are allowed to roam around. And the question was, could I have mapped the zoo in DNA? And I know a map of the zoo, so I can check myself. This is just one little sort of diagram of the zoo from the top down. Here are 7 different areas that we sampled and what we found in them. So this is where they keep the binturongs, and we found binturong DNA. That's a good thing. If you're in the donkey enclosure, you'll find donkeys, and the dingoes are in the dingo enclosure. So we're able to find things where we expect them to be. This is good news. But we found other stuff. So if you're over in the binturong enclosure, you get a lot of binturong, but you also get lemurs and tamarins and sloths and dinkos and howler monkeys. What you're seeing is DNA on the move. It's moving through the zoo. This is DNA that has blown from one enclosure into another one, and we've caught it. This is really good news. It means that I don't have to be in front of the animal to detect it. If I have to be in front of the animal to detect it, there's no point in doing it because I can see it. But if it can be hundreds of kilometers away or hundreds of meters away even, and I can detect it, I don't need to see it. It becomes a much more useful tool if you can do it at a distance. We also found wildlife. If you're in England and you tell someone you can detect a hedgehog, they get very excited. Hedgehogs, hugely protected species in the UK, very threatened. So what we're finding is these are animals that are walking through the zoo. It's an open area, the hedgehogs come and go, and we capture their DNA. So we're monitoring wildlife on the first try. We got ducks and some bats and hedgehogs and stuff that just bounds around the zoo. We also got stuff the animals eat. So in the binturong enclosure, you feed binturongs chicken, and the most predominant non-wildlife signal is chickens. So stuff they ate is in the area. It's probably the feces, again, being aerosolized. Over in the dingo cage, they prefer mammals, and so you get pigs, horses, and cows. That's what they're eating. So this was a really good indication that outside of that naked mole rat lab, we can still find DNA circulating. And if you think about a room like this versus all of the outside, that's a much bigger problem. Air is big. The atmosphere is enormous. And we want a tool that works outside. And this appeared to work outside. So we started developing other ways of doing this. We studied things like passive collection. This is like, I've designed a system that's like a little vacuum. It sucks, Could we do this without batteries? And so I actually teamed up with a guy who'd done plants in the air, and we used his little system to try and see whether these are dust collectors. It's really simple. It's a metal box with a sail on the end. And like a weather vane, it just turns into the air continuously. So it flips around, you capture all the dust, and the wind changes, it flips again. So every 2 weeks, we go and we collect the dust out of his little dust collectors and sequence it and see what's accumulated from the wind. This is probably the only real data slide I'll show you in the whole thing. This is 26 weeks, every 2 weeks for the entire year across the top. So each column is a different 2-week interval. This is a bunch of different animals that we detected. Green squares are detections and gray squares are nothing was there. You don't need to be able to read it, but I want to show you a couple of really interesting patterns. These are bunny rabbits. This is Texas. Bunny rabbits are really common in Texas, and they're really common in our data. So common stuff was common. If it was common in the landscape, we catch it all the time. This is good. This is the Texas toad. They're rare. We only found them once, which is good. Rare stuff is rare. We're trying to, at this point, just prove this technique tells you something useful. Rare stuff was rare, common stuff was common. This part of the year, there's almost no green squares. There's nothing being detected. This is when Texas is hot. There's really reduced vegetation. Stuff leaves. The other part of this graph has more green squares. This is when Texas is cooler. It's rainy, windy. It's mating season in Texas. All the animals come back. They come back into the area. They get really, really active, and we start to detect them. So this tells us that we're actually detecting real things that are happening at a relatively good interval. And probably the best one is actually that Texas toad. These things are really uncommon. They have a really interesting life cycle. They basically live in semi-dried out ponds until there's a massive rainstorm. They come out, they do a lot of mating, and then they go away again. The week before we detected the Texas toad, there was a massive set of thunderstorms. This is almost certainly thunderstorms, rehydrated a pond, the toads came out and got busy and then went back to sleep again. So an actual event triggering our ability to find the animal just with an air sample. I never saw any of these things. So we began building these little things. This is my one foray into engineering. This is a computer fan. It has a 3D printed grid on top. And if you think of a fan in your computer that turns on and blows, it's got to suck somewhere. So this is basically a teeny tiny little vacuum, sucking from one end and blowing out the other. And we suck in through this filter. This is actually a furnace filter from Home Depot. Furnace filters, if you read the packaging, say suitable to remove viruses from your air. Viruses are smaller than animal cells and therefore DNA. So we just cut up Home Depot filters and suck air through them with a computer fan. And this is the latest in technology. This is a string in a room in the jungle in Belize. Every one of those little white bags has a bat in it. This is how you carry bats around. They just go to sleep. So you go out to the net, you catch a bat, you put it in a bag, you bring it back. My job, I'm actually a trained taxonomist of bats, is to identify them all. So if you look in the background on this, there's my little handheld samplers. One of our first experiments was, I know all the bats that came in because I identified them all. We just filtered the air in this room for days to see if I could have identified all the bats without looking at them. Imagine it's a cave and they're coming and going in the same way that I'm bringing them in and letting them go again. Could I have sampled a cave this way and never even seen the bats, let alone catch them and pull them in bags? You know, it's a reduction in animal usage is what it is. And the answer was yes. So this is the number of DNA sequences we got. Each dot is a different species of bat. And this is the number of them we had. So some bats, we got hundreds of them. Some we had very few of them. There's actually a reasonably interesting correlation. The more we have in the room, the more DNA you capture. This is a fairly dangerous correlation to do because there's lots of things that throw this off. And I'll show you a good one, but the more animals we had, the more DNA we got. It's a pretty good thing. With the exception of these guys up here and this one down here, This one down here, we have far less DNA than we would predict for the number of animals we had. These are vampire bats. I don't take them out of the bags. If I touch the bag, it goes, I know what it is, I don't look. So I never took them out. They didn't shed much DNA. We didn't capture much DNA. These three up here are really, really hard to identify. I spent a lot of time looking at those. And my action of taking them out and examining them put lots of DNA in the air. We caught lots of it. That's why it looks like that. But we've moved on. This is one of my favorite pictures I've ever taken in the field. This is one of my students, Nina. She is sitting in a looter's tunnel in a Mayan ruin that is now used by bats. She is in some of the oldest engineering we can find with some of the newest engineering. She is setting up samplers to monitor bats in this tunnel. And we've monitored bats all over the place. These are more tunnels cut into the Mayan ruins by looters. These are natural roosts, trees, tree hollows. And we are now monitoring bat populations just by collecting air from inside the roosts. These are bats that we regularly see, so we catch them, we know they're there, we're confirming they're there with DNA. We have found new ones. This is a species of bat called a white-winged vampire bat. In 24 years of netting bats in Central America, I've never seen one alive. They're really, really good at evading our nets. Our very first try, we got their DNA because it's a different way of monitoring. So we can find missing species. We do lots and lots of different kinds of sampling. This is us lowering samplers into a cave to try that we can't access. We can't get in this one. It's too dangerous. You can put them down on ropes, collect DNA, and learn what's in the cave that you can't get into. Beehives. We're actually trying to monitor the health, the microbial health, and the plant visitation of colonial bees just by taking the air out of the hive. And we can track where they've been. But it doesn't really help me with this. So this is a picture I took in my field site when I was a graduate student. This is Costa Rica. And I am no better now than I was then at telling you what lives here. Because this is still one of those tools that works in a cave or in a beehive. It doesn't work at the level we need to measure. Remember, I told you that 90% of the world's biodiversity is unknown, that we have no monitoring system for measuring life at very large scales. I'm great at bats and belize. We suck at everything else. Trying to tell you what lives in Canada is an impossibility. I can't tell you that. No biodiversity scientist could. So how do we go to this? Remember the problems of biodiversity. We have no standardized methods. Everything is specific to the animal or plant you work with. We don't have any, because of that, we don't have any standard way of measuring stuff. And why? Because we have never invested in infrastructure for measuring biodiversity. If you look at the United Nations Sustainable Development Goals, they talk about needing to measure life on land and life in the oceans. If you read the Convention on Biological Diversity and the commentaries, people will say the biggest problem is that there is no infrastructure to actually measure this stuff. We've never built it. But it turns out we did. We didn't know it. Round about the time that we were doing the study in the zoo, the Natural History Museum of London wrote an article about what we were doing, sucking DNA out of the air. And it was read by a physicist. His name was James Allerton. And he called me or Joanne Littlefair, I can't remember which one of us got the phone call. And he said, I don't know anything about biodiversity. He hadn't been to my lecture. And he said, explain to me what you're trying to do. I said, well, we're trying to find new ways of measuring life. And he said, are you telling me all you want to do is filter air? I said, well, yes. And I built these marvelous little devices. My kids helped build them on the dining room table, and we can stick them in caves. But you know about 1 million of them to survey a forest, sticking one on every tree. And he said, you just want to measure air? I said, yeah. He said, have you thought about the planet's pollution monitoring system? I said, what's that? He said, I run it. So he is actually one of the people who operates the heavy metal detection system of the UK. When we realized that burning fossil fuels, running your car, produced pollutants, it was regulated to protect your health. There is a maximum you're allowed to be exposed to something like lead or arsenic, nitrous oxide, things that you get from burning fossil fuels. And once you limit exposure, you have to measure it. So we've devised a system of these samplers, which use a little filter like this. It goes in, it takes a sample every day or every 3 days or every week, depending on what country you're in, and it measures your exposure to these things. How much at risk are you of inhaling a pollutant at a level that's dangerous? And he said, we operate these things everywhere. They're sort of semi-automated systems designed to measure the environment's exposure to pollutants in the air. He said, is it possible that they have been collecting DNA and nobody looked? And we said, well, you better send me one to test. So you put them in, they look like this. They come out, look like this. These are all the particulates that have been attracted. Remember, I told you that inorganic rock and particulates traps DNA. We know this because DNA is sticky. So he said, is it stuck to this stuff? So he sent us these little triangles that he cut out of these filters. And he sent us 17 of them. Half of them came from the Scottish heathlands. This is rural areas of Scotland where they measure every week 1 cubic meter of air per hour at exactly measured regulated intervals. And then the filter pops out, a new one goes in, and it does it again. It's automated. So he sent us some filters. He sent another set for us from Bushy Park in London. This is not actually on the monitoring system that he runs. This is an experimental system we could use. He sent us some of these. And from 2 locations and 17 samples, we identified 180 different taxonomic groups whose DNA had been trapped by accident. And we looked into these, a lot of them were plants. That makes sense. Bushy Park in London is basically an urban forest full of stuff, ornamental plants from people's gardens, ducks and insects and mammals and stuff that was just there. It's all being captured on these filters. We looked at plants and insects and vertebrates. I'm not going to drag you through this, but I'm going to show you 2 important patterns. These are plants. So we measured for an hour of filtering air, a day, and a week. And the only thing you need to realize is that between a day and a week, there's not really any difference in the number of species of plant you accumulate. And we thought about this for a while, and it suddenly occurred to me, plants don't move. After a day, you have basically saturated, you've captured all the plants in the area, and sampling for longer isn't going to get you any more. If you're sampling, for instance, animals that do move, the longer you sample, the more you will find, because new things come into the environment, we capture them on the same filter. But there aren't just 2 filters in the UK, there are more of them. And so this paper we published about a year ago, and we said, the planet is basically covered in these things. The UK has probably about 150 of them. We only knew about the set that do heavy metals, but there's other ones that do other things. There are, I think, 3,000 of them in the US. There's 1,000 of them across Europe. Canada has about 250 under different regulatory schemes. They're all doing this. They're all taking air filtration samples every day or 2. Each one of them we think is capturing DNA. We found them in the Amazon. We found them in Peru. We found them in South Africa. They're lining the Amazon forest in Brazil. They're everywhere. And they are a pre-existing infrastructure. They've been around since the 80s to measure our exposure to pollutants. But as a side effect, they are capturing DNA in a massive survey system that nobody had ever seen before. Can I ask you to turn off your recorder now, Daphne?